name is Dan Macqueen. I'm a full professor based at the Roslin Institute at the University of Edinburgh. I'd like to thank the organisers first for accepting our abstract for an oral presentation in this workshop. And as the Deputy Coordinator of AQUA-FAANG, it's my pleasure to provide this high-level overview of the project on behalf of the whole consortium, including the lead coordinator, Sigbjorn Lien, where we're aiming to functionally annotate the genomes of 6 farmed fish species of key importance in European aquaculture. As you'll be aware, aquaculture is a critical sector for global food and economic security that's growing very fast, with more than half of all the fish that we eat on Earth now being farmed. Farmed aquatic species are genetically highly diverse, as you can see in this phylogenetic tree taken from this review that was led from the Roslin Institute. And aquaculture species are also much less domesticated on average compared to livestock and poultry. And that recent domestication history means there's a lot of genetic variation that's available to be exploited towards genetic gain and trait improvement in breeding. Another important thing to note is the devastating impacts of infectious diseases in aquaculture, which has been estimated to cause losses exceeding 20% of all production globally. This causes major concerns for animal welfare and also for disease transmission into wild populations, and it's a really big challenge for the scientific community to address. Genomics and functional genomics already plays a key role in aquaculture research, informing our understanding of the biology and genetics of aquaculture species, and then it's also, of course, critical to exploiting such knowledge. All the major aquaculture species have annotated reference genomes of different qualities that are being widely utilized by the research community and by industry. However, to date, the functional annotation of these genomes is almost exclusively limited to RNA-seq evidence, and we have a poor overall global understanding of many regulatory and epigenomic features in farmed fish and shellfish genomes, but this knowledge is building up quickly. This figure is effectively emphasizing something which I don't think I need to convince this audience of, that genome functional annotation is central in genetic studies, for example, to be able to help prioritize potential causative genetic variants which can be used in aquaculture breeding. AQUA-FAANG addresses the gap in annotation of farmed fish genomes in addition to the disease problem that I highlighted earlier by generating genome-wide functional annotation maps for the 6 species shown in this tree, which together account for over 90% of finfish production in European aquaculture. And we want to be able to exploit the contribution of functional genetic variation identified in the project to commercial traits with a focus on improving resistance to disease. This is a €6 million project funded by the European Commission Horizon 2020 scheme under the same call that funded the Bovreg and Gene Switch projects. So the concept of AQUA-FAANG is to improve the functional annotation of these key farmed fish species, with the overall objective being to have positive economic and sustainability impacts on the aquaculture sector, ensuring the results generated are being used in practice, particularly for genetic improvement in disease resistance. To achieve this end goal, we have 5 specific project objectives, and I'm not going to actually go through these individually now in the interest of time, but I briefly explained them in the accompanying poster recording for those that are interested. AQUA-FAANG has participants from 8 different European countries, including a range of leading academic institutes, many of which are well-established contributors to the broader FAANG initiative. We also have industry partners on board that are spanning the different project species. These are mainly genetics and breeding companies who are interested in exploiting the functional data generated. If you'd like to find out more about our partners, then please see our project website where there's more details available. Just very briefly, AQUA-FAANG is organized as a project into 6 scientific work packages. I'll briefly touch on in the following slides. We also have a very dedicated work package 7, focused on communicating the project and disseminating and exploiting its results. But I won't say much more on that today. I'm going to focus today on the scientific work packages and the progress that's been made. I also wanted to, before doing that, promote briefly Eurofang, which is an important achievement for the 3 farmed animal genome annotation projects that were funded under the same Horizon 2020 call. This initiative was born out of a meeting which occurred just before the pandemic at the Hinxton campus and was coordinated by EMBL EBI. And EMBL EBI have been playing a leading role in the Eurofang initiative, which really represents an effort to communicate effectively and share knowledge and expertise and coordinate a range of activities, including for communication and dissemination, in addition to collaborations and training across the 3 projects to really sort of drive forward the impact of these projects very broadly. More information is available online if you'd like to find out more about Eurofang. An important point to note on genome sequencing is that greatly improved chromosome level reference genomes with, as illustrated here in red, hundreds of times increased base level contiguity have been generated during the time course of AQUA-FAANG already, and partners in the project have been playing a leading role in generating these improved genome resources. So that's for the species that have been highlighted in blue. These improved genomes will be used for all functional annotation that are performed in the project, which is being done using standardized pipelines coordinated within Work Package 2, which is led by EMBL EBI. And as part of Work Package 2, these new genome versions are being uptaken to the Ensemble Genome browser, where all functional annotation in the project will be made available, and the genomes themselves will be made available in full for the first time in the 105 release, and in advance of that on Ensemble Rapid release. Coming on to the work being performed, I'll just quickly run through some of the key data sets being generated in the project. And I'd like to firstly, before that, emphasize that a huge amount of fundamental work has gone into the project in terms of developing reliable functional annotation protocols, as methods like ATAC-seq and ChIP-seq have not been widely used in aquaculture species to date. There's been a lot of optimization across different stages and tissues, really from the ground upwards with these approaches. And within Work Package 1 of this project, which has been the hub for such groundwork, we are also performing a rich functional annotation spanning the lifelong development of the different species from embryos to adults. This includes what we call dev maps, where we're defining regulatory elements and expressed features across standardized embryonic landmarks in the different species, starting from when the zygotic genome is being activated through to when the tissues and body plan have been fully patterned in the larvae stage of the fish. And this work is now well underway, with most of the samples already obtained and most of the RNA and ATAC libraries sequenced. And ChIP-seq's already completed for 2 of the species and in progress for the others, so we're approaching having 500 sequencing libraries for this work. So our functional annotation of post-embryonic stages of fish ontogeny are focused on a panel of tissues common to the different species at 2 stages and in both sexes for both juvenile and sexually mature fishes. And these have been defined according to the status of the gonads by experts in the project in reproductive physiology. Again, we've made very good progress in the work. The sampling has been largely completed across species, and over 1,300 sequencing libraries have already been generated and analyses are beginning. In Work Package 3, we're performing functional annotation to understand responses of the genome to immune stimulation across the species using a sort of standardized approach which is comparable across the species. And this is to use immune mimics or PAMPs to stimulate either the antiviral or pro-inflammatory response in these species using poly-IC and inactivated Vibrio bacteria respectively. The samples are from head kidney, which is the primary lymphoid tissue, or from leukocyte primary cultures that have been taken from head kidney, and the samples have been selected on the basis of marker genes across a time course post-stimulation, which has led to an effort to standardize across the species. And this sampling has been largely completed, and the functional annotation work is advancing fast, and as you'll see, more than 400 libraries have been sequenced to date. AQUA-FAANG also has a work package that's targeted at exploitation of functional annotation data with the aim to understand the mechanisms underpinning disease resistance and enhance the accuracy of genomic prediction for disease resistance. There's a lot of activity in this work package, which is being led from the Roslin Institute by my colleague Ross Houston. This includes a major genotype to phenotype viral disease challenge study in sea bass, with over 1,000 challenged animals that have been genotyped already to whole genome level, and a subset are currently being used for RNA-seq and ATAC-seq data generation. The RNA-seq data will be used in an EQTL type approach, and the ATAC-seq data will be used to help prioritize variants in QTL identified. And further to that, functional annotation of disease resistance QTL that are already known will be performed in several AQUA-FAANG species using existing or new challenge experiments. Later in the project, there'll be efforts to test models or test whether models incorporating prioritized functional markers improves genomic prediction accuracy, and also developing with their industry partners functionally enriched low-density marker panels for cost-effective genomic selection. And both these approaches that I've emphasized at the end are in demand to a different extent depending on the species sector. Final scientific work I'd like to emphasize is in work package 6, which is comparative analyses across the functional annotations performed in the different species. The rationale for this work, which is really in early stages because it's very much data dependent, is that comparative approaches provide a more informative annotation by defining the extent of evolutionary conservation of defined genomic elements. Of course, that's then linked to functional importance of those elements, which has applied and transferability value, including to different species outside the project that are related to the species in the project. And importantly, comparative analyses will also help us understand duplicated features that are retained in the genomes of these fishes from the whole genome duplication events, which have been highlighted here in this tree on the left. So there's 3 whole genome duplication events in our project, and we're very interested in the functional importance of the widely retained duplicated elements in these fish genomes. So I'd like to finish there and thank you for your attention. I'd just like to acknowledge the incredible set of partners in this project that have really driven things on and made great progress in spite of the global pandemic, which has obviously had lots of negative impacts. I've not been able to mention all the partners by name who are leading different aspects of work, and there are just far too many individuals to thank, but I would like to acknowledge broadly everyone who's been involved in this really nice project. It's been a pleasure to be part of it.